One. Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in previous life I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC dedicated in the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but since then I've worked as a research integrity specialist, and now I'm currently working for a scientific journal.
Every week we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen during the week.
Yeah, and at the end of the month, well, in the middle of the month, we're going to be looking at a paper on bioprinting. We're definitely working on it, but obviously we want to do more deep dive. So if you have any idea for something that's really captured your interest, please let us know through Twitter at microtwjc.
Yep, you can follow along with the papers that we discuss on all our weeks in our shared Zotero Library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And boy, do we have a show for you. Firstly, we have a deer. What kind of a deer? How's that got to do with SARS CoV-2? Like, where are the deer getting infected? We're going to do some deer research in SARS CoV-2. But not only that, we've got antigen testing. So can we predict how contagious SARS CoV-2 is based on a small test? Or what else? Even can we predict how severe the next variant is going to be? And what other things are there? We've got a summary of what's up with Omicron. It's been like 4 months. What do we know about it? Also, SARS-CoV-2 in the liver. What's it they're doing there? We'll find out. Then we've got our non-COVID-2 SARS-CoV-2 related papers, which are CRISPRs. Something new about CRISPRs. What other things are there? Oh, mitochondrial transplantation using single-cell surgery. That's going to be a fascinating one. And asymmetric. Tiny
Peptidoglycan curvature.
Yeah, so this is about like curvy bacteria that eat other bacteria. How do they do it, and why are they curved? We're going to find that out as well. And finally, how do you like dem apples? Candida on apples. Are apples a secret of why we have Candida auris? All that and more. Stay tuned.
So yeah, as per usual, jumping into SARS-CoV-2 things, this is a real short paper, serological evidence that SARS-CoV-2 has not emerged in deer in Germany or Austria during the COVID-19 pandemic. And yeah, so this is, I mean, it is what it is. It's quite a simple study. They use like a pseudovirus neutralization assay to try to see if these deer have been exposed, so they can take some blood samples from deer and then check whether or not there are any antibodies in that blood to neutralize a pseudovirus, right? So that's like a viral particle that's covered in SARS-CoV-2 spike protein, but not like inside is not SARS-CoV-2. And I think this is pretty similar to the technique that's been used previously to show that deer in North America do have a high amount of antibody towards SARS-CoV-2, but interestingly, in this paper, they find that the deer in Germany and Austria do not.
Yeah, so I mean, we've been talking about this a while, where we've looked at white deer specifically, and we know that like in North America, white-tailed deer do carry SARS-CoV-2. Yeah. Sorry?
No, I was gonna I just want to say white-tailed.
Yeah,
the Bambi, right? The Bambi of deer.
Yes. Exactly, and we've known because there were like a couple of like surveys done by hunters who were swabbing their kills to check whether they had SARS-CoV-2, and they found that there was some interaction between them. But so naturally, these researchers were going to Germany and thinking, well, we've got some deer populations, and we've had COVID. Maybe our deer have had the same level of infection. Yeah,
totally. I mean, these are different species of deer, but presumably, like, whatever that ACE2 similarity is between these deer and our ACE2, it's like somewhat similar, and that's why like you'd expect that these deer would also have virus inside of them. But they do not, and so that sort of draws out like a whole bunch of different hypotheses from the authors, mainly focusing in on just, I guess, the culture of hunting and how deer are managed in in Europe, and citing that those differences may be why they see, no spillover into this
Yeah, because
population.
deer hunting, it just isn't as big in Europe as it is in America. I think there's a lot more like, I mean, I guess Europe did have a culture of culling deer, but I think we may have like just gone through all the deer and now we're not allowed to anymore. Um, I'm
Yeah, they're also maybe saying like there's not a lot of. It sounds like what they're trying to say. They say lack of urban or peri-urban populations of deer. I
right.
seem that's sort of like suburb. I guess like deer populations that live near cities or in suburbs, something like that.
Yeah.
I guess that doesn't exist either in Europe. Like it's like strictly the rural deer, not like close to high density population deer.
I mean, that sort of makes sense. I imagine that like, because I hear in America you have deer raiding your trash cans occasionally in like certain areas.
Yeah, definitely in some burbs, I think that people like, yeah, suburbs that like encroach into forests. I know that like, yeah, deer kind of can come into the burbs. Like that could be one of the things. Again, they don't really have like a definitive answer on this front, but they're trying to explain, right, through the differences in wildlife management between the countries, like what might be contributing to this, lack of
Yeah.
SARS-CoV-2 in the deer. They also say something about like bait trapping, that like bait trapping isn't as practiced inside of Europe, but like it is in the States. So like it could even be like just, the action of bringing deer together with food that was touched by humans or that humans are distributing. Like that's like a
Right.
contact point.
Yeah.
They don't have a definitive answer on this front, but it is interesting to think that I mean, maybe it goes back to some of the things we've saying before. Like behavior does matter quite a bit in like determining the network of transmission that we have, and it can even impact. Like basically, if you isolate from the deer, then like the deer won't get COVID. There's that's interesting from that
Yeah, this is a
perspective.
very short paper. It's very much like they're very much a minimum publisher unit kind of paper. Where they got like one or two graphs, but there are maybe a few
Yeah,
extra questions, like because you can't answer with this data. I don't think they've really answered whether these deer are even susceptible to SARS-CoV-2. We don't know that from based on this,
yeah.
and so
Absolutely. Yeah, so like there could be an underlying thing, right? Where even though like evolutionary speaking, these deer are similar to other deer, right? They might have something that also like makes them less susceptible, like ACE2 or whatever, from getting
yeah,
SARS-CoV-2.
so it's an interesting sort of study, but very short, very like concise. So we can move on to the next
Yeah.
one, which is also kind of a quick paper, which is talking,
Yes,
yeah,
yes.
usefulness of antigen testing in predicting contagiousness in COVID-19, which is something that's kind of very like relevant for people like me, where I've got like which I have to make sure, yeah, to test
Yep.
whether I'm suitable to be allowed in society. Um, on the basis of being with COVID, not just
Yep.
generally.
Oh, they don't have lateral flow kits for like how
Not
sociable you
yet.
are. I picked this paper because I feel it's quite practical, and like previously, on a personal note, right? Like back at the beginning of the Omicron wave, we had Omicron in our household, and I was like, oh my gosh, like how do we exit this scenario where I'm isolating from the person I live with? And I like pulled a bunch of research, and people were talking about, like how many days, like they call it DSO here, days since symptom onset. And I was like, well, that's basically information that one has typically, right? When you learn that someone in your network has SARS-CoV-2, or when you have SARS-CoV-2, like this is a piece of data that you have. And then you're saying, okay, well, how many days do I have to wait? Do you just wait like the 2 weeks, right? And like that's sort of like the safe time or whatever, or is there something like testing we can do, right, to figure out if like we're ready to re-enter? And of course the most important thing that we want to look at is like, are you contagious? But I don't know if folks remember or recall this, but like PCR is super sensitive, which is great for finding it even before you have symptom onset. But it also means that at the tail end, you're also still testing positive even after presumably like your infectiousness has gone down. And so in this paper, they compare the RT-PCR test to that lateral flow antigen test to viral culture. And of course, viral culture being like the most important readout, because that's like actual virus coming out, being able to infect a cell culture.
Yeah, it's interesting because RT-PCR can pick up, because there are like RNA that hangs around after the infection, which doesn't produce infectious viruses. So RT-PCR can pick that up. So it can pick up like previous infections, but it can't necessarily. But I think like it can be oversensitive from the perspective of someone who wants to get out of isolation.
Yeah. Yeah, like at the end point, right? Like I still feel like RT-PCR has this like really cool ability that like if you have like a really high risk exposure event and then you're not showing symptoms, but you want to find out whether or not like it could have been SARS CoV-2 or you could have been exposed to SARS CoV-2, like PCR tests fit, I think, that niche really well at the beginning of infection. But like kind of like during and at the end, like it, at least based on what the paper shows here, it seems like antigen tests do give a really good sense of whether or not you're going to be viral culture positive, which again is this measure of like actual virus coming off of you.
Yeah, I noticed like some behavior these days of like people I know who would basically go off whether they can go to some event based on their last negative test rather than waiting the full 2 weeks. And this kind of like shows the validity in doing that. And how like, at least when we think about having to live in a society with this virus. So we know when we're infectious and we can isolate based on those tests.
Absolutely. Yeah, and they go through some like positive predictive value, negative predictive value in terms of like, like are these tests picking up? How often do they miss something, right? Versus how often do they catch something? And yeah, those values look good. So lateral flow tests are very good tools to be using. If you have them available around, just
not sponsored.
Incorporate them as
by lateral flow tests. No, but they have started charging for lateral flow tests in the UK because we live in a hellhole. But if they want to send us more, then this would be great. Just
well, Foz, they've always charged for lateral flow tests in the States. And I mean, you can find a bunch of you can go out and get a test, right, at a clinic or whatever, and like they'll charge your insurance or whatever. And then the federal government has issued like 4 free tests. So yeah, welcome to the hellhole.
Okay, right. Okay, fine. That shows me for trying to complain about anything to an American. That's certainly right. I should lessen. It's just like, okay, if I have anything to say about like healthcare or not,
but I will say like, but I'll say like again, like maybe sharing a bit of more of a personal story of my experience with SARS CoV-2, that it is nicer to be in an environment where I can actually buy tests at the grocery store versus at the beginning of the Omicron wave where like there just weren't any tests. So at least like, we're given these tools. Yeah, we got to pay for them, unfortunately, but they can really come together to give us confidence that we're managing infections as they arise, like within our personal networks.
I think that brings us to the next paper, which is an interesting one. So it comes from inconsistent directions of change in case severity across successive SARS CoV-2 variant waves suggests an unpredictable future.
This is great because I feel like we had some comments in our in our chat box a while back, like sort of bringing up this point that there's been like inconsistent severity across some of the emerging waves, but now we get to see some graphs that show the same.
So they kind of ask the question, they compare waves to ask the question, okay, can you predict what the severity of the next wave is based on past predictions? And so they do comparisons of the original Wuhan strain with the B.1.7 alpha variant, and so on. So you get some.
I think it seems like the way that they do it is they're like comparing concurrent strains. So like at every wave, there's been like a new thing taking over. So they're looking at the cases that are happening sort of around the same time, but are coming from the new variants. The old variant versus the new variants, at that.
I think
like,
that's a really important thing because sometimes these breakpoints have other factors that are at play, like the level of kind of protection, like the level of like kind of, I was about to say sanctions, but
vaccine, right? Like vaccine things have gone up, so like you don't want to necessarily just compare like the latest variant to a very early variant, because like there's a lot of covariates that have shifted between the two. But this graph with the little yellow bars, I feel like that's the one where I feel is like trying to illustrate this point, that they're only looking at like one variant versus another variant when they were both infecting at the same time in a population. So that base population is kind of the same for all intents and purposes. And they split it out here by age group.
So in general, like it seems that with each variant, from the original down to Delta, it seems that it's been getting more and more severe.
Well, the original down to no, Omicron's not as severe as Delta. Right?
Omicron is the thing that breaks it's where Omicron is the one that's less severe than Delta, and this study doesn't encapsulate how Omicron relates to the previous strains. So I think there are other studies who've looked into that.
And they also have AY. I think we talked about this one too. I don't quite remember. Like it hasn't been in big circulation, right? AY.4.2. That one also seems like it was less severe in Delta during the time they were together.
I think
but Delta's more severe than Alpha.
So you'd have no idea of which one is better or not, and it does seem like the majority of them seem to be better. And AY 4.2, that was like the almost next strain. It was so Omicron kind of stole its crown for being the next strain.
And Alpha was more severe than B1.177.
Yeah, that's right. So each variant has gotten slightly and you can't predict.
No, it's kind of like there's no clear pattern to say, right? Like with a variant, whether or not it was gonna contribute to more severe disease or less. Of course, like in our most recent memory, Omicron, right, compared to Delta, that has been less less people died or were admitted to the ICU. They don't even use the word severe in this, right? Because like I think that like that can get right. It really depends. Like severity gives you like is that sense of like you need to know more about the disease state. Here they're just saying like they die or they admit to ICU, which are ostensibly quite severe outcomes.
Another point is that Omicron is like just way more transmissible. So I think and again, there was no way to predict how much more transmissible it would have gotten. And that is another factor, because even though Omicron, like as a percentage, is less like lethal as a whole, we're still seeing like lots of like high levels of people getting hospitalized because of Omicron, and even though it's less severe because everyone's getting it. And I know recently in the UK we've been getting an increase in reinfection rates, which could be due to like spaces between last vaccination and last booster. So there's a lot of people who think that this outbreak is over. I don't think so. And I think this kind of hits on that kind of same. We're not at the level where we can say we're prepared for the next variant.
I think we might have touched on this before. I tried to pull data. Like the CDC had said that like during the Omicron wave, like so many more children experienced disease that sent them to the hospital. Hospitalization rates during the Omicron wave, even though right like it's not as severe for children, but like the number of cases kind of like drove it to that point. So I mean, even like, so we've talked about that before, that like differences in like getting sent to the hospital or overall mortality, like that can still like be a really bad thing if like tons of people get infected. But like I think that this paper like is speaking more towards just like right, there was a sense that because Omicron was less severe, so then that's like in line with this hypothesis that viruses are trying to adapt to us and like they don't want to kill us, which like might be something that is real. Like that's again like just a way of speaking about evolution. But like here in the evidence, like at least from the different variants that we've seen that are concurrent, that doesn't seem to be the trend.
Yeah, there is this like old like white wives' tale that says, "Oh, a good virus never kills its host."
Hmm.
I think like we need to add that never kills its host before it infects the next host. But
Yeah, that's definitely good. Also, when you say that that's an old wives' tale, it's like, right, it's like an evolutionary theory. If the old wives were like,
some
yeah, I mean,
evolutionary
not to
theorists
say
were
that
old
old
wives.
wives
Okay,
can't be, but
let's
they
not
were
be
definitely like,
exclusionary.
yeah, exactly. Yeah, exactly. Yeah, like I love old wives' tales. Like they aren't necessarily a
Yeah.
bad thing here. But I don't think they necessarily capture the reality, right, and the nuance that might be emerging. So like it's nice to have this data in front of us to definitively say, right, like no such
Yeah.
pattern.
Let's see the
Hmm.
next paper. This is an interesting one. So, selection analysis identifies clusters of unusual mutational changes in Omicron lineage BA1 that likely impact spike function. Um, yeah, so this is a paper from like a couple of people we've done a lot of. I mean, mostly like people who've like been quite active in talking about Omicron on Twitter and on other kind of mediums. So I remember like Tulio de Oliveira was a person who kind of like gave this big talk about what Omicron was doing in South Africa that spread around the internet. But now finally, after about I think must be like 6 months, we're finally getting a paper that summarizes all those early findings.
Yeah. Yeah, so I guess the question, right, is where does it come from? Because like we saw like all those different mutations and trying to, I guess, bin the mutations in different ways and look at how those three lineages, the BA1 and BA2, right, which I think is driving the waves in a lot of places now. And there's another one, there's
Yeah,
BA3. But being able to compare like the clusters of mutations that are occurred on these lineages to try to trace back where they all came from.
yeah. They talk about how I think 13 of the new mutations have been kind of seen previously, and then they look at
Hmm.
the like other mutations that have appeared. The main conclusion they come to is that a lot of these mutations are can only happen because of the other mutations. That like
Mm-hmm.
I think it said about like I think 16 mutations. Actually, no. Let me double check this because I want to get my numbers right. Uh, but
Yeah.
actually, I'm going to say like roughly about half the mutations are things that would be detrimental in previous strains, but now appear to be
Yes.
actually
Yes.
adaptive in this strain. So enough mutations have happened that they've kind of stacked up on each other. Like they like
Hmm.
you've gone down the skill tree in your kind of RPG.
Yeah, another thing that they talk about, like, so they don't come to any conclusion as to where Omicron comes from at the end of this paper. In some ways, it's just it's catching up the story, right, of like where it could come from. What are the hypotheses that are being flung around? So, right, one hypothesis is that it comes from unsupervised mutate like community transmission within an ungenomically surveilled population, right? And then and then when these lineages did become successful, they appeared right in South Africa, as we know, and the surveillance caught them then,
Mm.
right? And it was some other community transmission somewhere else that caused all this diversity to appear. But then there's also the idea that what if it was a zoonotic event, right? This is like it was in an animal population again, and then it came back into us, right? That there's this hypothesis that lies around, and that's like one of the like the idea of the neutral mutations, the deleterious mutations, like that's part of the evidence that they could say, oh, maybe some of those accrued because they were in a host that the pressure wasn't the same. also along the line of like being evolved in a place where the evolutionary pressure was different, the hypothesis that it emerged within an immunocompromised individual, right? There was like an immunocompromised patient 0 that like harbored a bunch of diversity. So these are all hypotheses, and the hope was that going through the mutational data, they could try to narrow it down a little bit, but at the end of the day, they sort of say we need to know more. They don't have enough just by looking at the current snapshot of mutations to differentiate between any of those 3 hypotheses.
Yeah, that makes sense. I mean, there's always a little bit of mystery that surrounds whenever a new variant comes out, because I think, like, again, we're not quite certain why, like, I mean, the Omicron's getting a lot of attention because it's so different, so it's hard, we literally don't see the steps that came to make it, whereas with kind of the Alpha variant, we saw that those steps come towards it because it emerged in the UK. But in India with the Delta variant, there is a bit of like, what, how did that come to be? So yeah, 6 months later, we still aren't necessarily sure about the origins of Omicron, like how it happened. We've got some ideas, but
yeah, which sucks in my mind, because it's nice. It would be nice to know. Okay, I'll say this. Like, I kind of just think all 3 hypotheses, we should just think of them all as being true, because they all imply like a way of dealing with the pandemic globally that like we should take seriously, right? Like, we should be preventing zoonotic transmission. We should be preventing the infection of the immunocompromised, right? Like, these are all like important like lessons in some ways. And so yeah, like I'm almost like, we don't need to find the answer. Just the action of talking about these hypotheses sort of implies like different actions that governments can take, or like that communities can take in order to ensure that we don't have any more variants. And of course, like the biggest one around this would be like the global vaccine equity, right? Like, we need more vaccines around the world to help like just reduce the transmission events and like restrict the space that SARS CoV-2 can play around in order to find these jackpot genotypes that then can like spread into populations.
Yeah, for sure. I think does that bring us to the end of? No, not quite. We've got one last SARS-CoV-2 paper.
One last SARS-CoV-2 paper. This one, the molecular consequences of SARS-CoV-2 liver tropism. So we've talked a lot about how SARS-CoV-2 just isn't solely a respiratory pathogen, that there's all the reports of finding it inside of the heart, causing issues in the heart, in the kidneys, in the brain, and now the liver is being added to the list here. I think in previous reports, we've seen like a mix of like a human-based study that shows SARS-CoV-2 in certain tissue types, as well as the matched animal model that comes along with that, right? With the idea that like once you find the signature in humans, you can then find an animal model that correlates and maybe do some research to figure out how to deal with this. This paper doesn't have the animal component. It's really just figuring out in humans, right? Like trying to make that argument that SARS-CoV-2 is doing something in the liver. So actually, Fuzzy, if you could go back to that figure F1F. Yep. I think that's like a good takeaway from the beginning of this analysis, like arguing the reason why they went down this path is just this one liver measurement they have, ALST. It correlates with the mortality of SARS-CoV-2 infected individuals. And so there's this idea that like, okay, like if there's like wonky stuff going on in the liver, like the outcomes are not going to be good for your infection. And of course, that could just be because the disease got so advanced. So that's not that you can't use that as sole evidence to say that like SARS-CoV-2 is infecting the liver, but it sort of argues for the relevance of like looking in the liver, and like it adds that little clinical importance to all of this, right? By understanding the effects on the liver, we may come to an understanding of SARS-CoV-2.
Yeah, I mean, liver is an important like aspect. It does a lot of work cleaning up the blood. There's a lot of immune cells there. There's a lot of plays a role in sepsis. So if someone already is like septic, then you're probably going to get viruses showing up there, and if they're causing damage, that can be an early signal of that kind of bloodstream infection.
Totally. But I think like from that same rationale, you could also say that like that's just the hallmark of a disease, right? And so like you really want to pin it down to SARS-CoV-2 as like it has like an affinity for the liver. And so then they go in the next set of diagrams, they like look at the ACE2 levels in the liver,
Right.
and they look at like spike protein in autopsied livers. So again, this is like typical workflow of what we've seen in this type of like expanding our understanding of where SARS-CoV-2 is infecting. They just got to link it to the tissue type, right? In real infections, show that like there is the possibility for SARS-CoV-2 to get in there. It is in there. And then what they can do is they like just do like transcriptomic analysis to say like, then what are the genes that are getting turned on during this infection? And like if you compare that to people that are dying from non-SARS-CoV-2, right? Or you can even compare that to SARS-CoV-2, but not in the liver, right? So like it'll give you a sense of like what is, I guess, specifically turned on in these disease states. And that could be a signature that either we choose to model in animals or use to, you know, suss out the differences in human infection.
Yeah, I think.
Yeah, and then to wrap it up, so they find differences, right? Okay, so like long story short, right? Like there's a lot of genes that change, and for me, what I really enjoyed about the paper is in Figure 4.
Ooh, okay.
So the figure after this is they take that signature, right? The signature of genes that are getting turned on during this infection, and they compare that to the signature of HBV, and HCV. Hepatitis,
yeah.
Which is a liver infecting virus, right? And they say, okay, and here are signatures that show us that like it's quite similar to another liver infecting virus. So, you know, like, yeah, the liver, like in disease, all your organs the organs can be affected in many ways, right? Metabolites change, and that's going to change how your organs work. But what I appreciate what this paper did was it also then linked the specific, I guess, like gene regulation changes that are happening in a SARS-CoV-2 liver infection to other liver pathogens, liver viral infections, and kind of being like, okay, well, this also kind of gives you a sense that it is doing something in the liver that is maybe liver specific. Yeah.
Yeah, I mean, this is. I wish we had like a doctor or a medical physician on this show, just so that I could ask the question of like, is this going to be quite useful in the diagnostic section? Because like from that first like graph in figure, I think 1E, where they had that, where it seemed like that
there's like
if
it gets in your liver, it's like predictive, right, of a bad outcome.
Yeah, these these AST tests are really like predictive of a potentially bad outcome. So could it be quite useful in that setting to find out who's going to have a more severe disease early on, and then maybe take steps earlier to try and prevent them from.
I sense, I mean, again, no medical profession here, right? Like, but I sense that the way that this paper is actually telling scientists things that doctors already knew, because I assume that, like I think doctors they take a panel of tests, right? And they by their practice, they start trying things out. Like maybe we should be looking at this level and like doing a different, like escalating this particular person instead of another, right? Like doctors have a very, it's a tricky job, right? Because like there's no 100% flow chart that exists, right? That tells you how to deal with every patient. But through their practice and through the extended infection, right? The extended course of this pandemic, doctors have converged onto different like flow charts essentially, right? To help sort patients. And we have seen improvements in the treatment of SARS-CoV-2 infection, right, over the course of this pandemic. But yeah, like, so like, I bet like doctors have like an intuitive knowledge of some of this already, but this is just like showing it to scientists and giving us like the charts and things that we need to then like go into other data sets and start like either making a model or like doing other like predictive things that are like fancy. But like, I would say that, I don't think doctors are learning too much new from this type of paper. They've already learned it.
Oh, yeah, that makes sense.
Uh-huh.
Okay, well, that brings us to the end of our SARS-CoV-2 coverage, but we've got some incredible non-SARS-CoV-2 papers. So we've got
Mm-hmm.
this one is quite interesting one. RNA-targeting CRISPR-Cas13 provides broad-spectrum phage immunity. This is another new CRISPR research from Duden Lab. They won like a Nobel Prize for CRISPR. And
Mm-hmm.
so CRISPR-Cas13 is an interesting one because this is an RNA-guided RNA nuclease. It has an RNA template that binds to other RNA. So it's been used for knockdown studies. It's not been
Mm-hmm.
used as much as I understand for genetic manipulation as the other Cases.
Right.
So yeah, it works like most other Cases. It has like a template and like a template called a CRISPR, which it uses to find like foreign like RNA,
Mm-hmm.
and then uses that to kind of signal to the rest of the cell that whether it's infected with a virus, and then it can either shut down the cell completely, or actually that's what it does. It chews up all the viral RNA, and then it just brings the cell. It kind of tells the cell to kind of stop doing everything so that it doesn't affect other cells.
Mm-hmm.
So what they did here, they decided to investigate it by looking at how common like the common variants of Cas13 in out in the wild. So there are like four flavors of Cas13. There's A, B, C, and D, and they kind of differ in the primary sequence and their size,
Mm-hmm.
as well as the other stuff in their system that's associated with them. Um, so they looked through a bunch of like genome taxonomy databases, NCBI, to look at how many of the different types of gene are out there, and so they found like that there's like a very like rare strain like Cas13a tends to be the rarest,
Uh-huh.
and they did a bunch of like tests on it and found it was actually like super potent. Uh,
Yeah, so it
uh,
comes so like I think we touched on Cas, like there's a lot of diversity, right? Like bacteria are a very diverse group, and like
mm,
this is a category, right? Cas proteins are a category of protein that exists across all these different types of bacteria. They
mm,
focused on one in their paper mostly, the LBU.
yes.
So like they're choosing like one specific Cas13 Cas13a from a specific bacterium, and it just seems to work really well. Like I guess it finds its target well is
Mm.
what they're why they've chosen it. And yeah, and then the story kind of says how they think it could be useful given this property that it can quickly recognize phage DNA. DNA or RNA?
RNA. So
RNA.
here's the thing: they what they found was that since it works against like DNA viruses as well, which kind of makes sense because even DNA viruses produce RNA in the process of replicating, and because they still need to translate proteins and other functions. So
Hmm.
the fact that this system can react to any foreign RNA means that it can still stop DNA viruses because it doesn't just act on the RNA; it acts on the whole cell. So
Hmm.
once it
So
gets
it
infected by a
doesn't
DNA
destroy.
virus,
Yeah, it doesn't destroy the genome of the virus, right? It destroys
right?
the proteins that the virus is trying to make off of its DNA genome, or
Yeah,
the potential to make those proteins.
yeah. I think it also it like chops up a bunch of other like so it's a non-specific RNA polymerase, which means that it doesn't just chew up the phage DNA, it chews up all the sorry, it doesn't chew up the phage RNA, it chews up all the RNA, basically kills
Mm,
the cell.
mm,
And
hmm.
that is really an effective way to fight a virus, not for the cell itself, but for the
Yeah.
rest of the community. If that cell's removed, then it can't spread virus to other cells.
Yeah. Actually, this is really similar to the defense strategy that we talked about in our last news show, where we talked about the defenses that were on satellite phages,
Yes.
right? Like those tools, they like it's like upon discovering that you're infected by something, you just kind of shut down everything. You trigger some sort of pathway that like suppresses everything because that's gonna make sure that that phage doesn't continue, because yeah, through the host, right? The host is essential for the phage's life.
Yeah. And so the weird thing is they found is this was kind of rare, and so
Hmm.
so I thought maybe there's two explanations. Either it's not effective, or it's too effective and can cause the cell's problem if it's activated just by non-lethal RNA.
Sure.
So I don't think I think they did come to an answer with that. They basically find out that it's very effective as an anti-phage system. But they didn't really. I don't think they feel like they articulated how it really harms a cell.
Sure. Yeah, that's their hypothesis. Like, they see the observation, it's rare, and then they're bringing in this hypothesis, right? That like there must be maybe there's a trade-off, right? Some evolutionary trade-off, right? Like, yeah, you get increased protection, but maybe you're less flexible in different areas. But I guess in true CRISPR fashion, like then they say, how do we use it for our own purposes? And so, yeah, unsatisfied with, or like just like not going down the story of like what it is in nature too much. They then say, oh, well, the ability to recognize phage infection and really prevent certain genotypes of phages from reproducing, this could be a really great way to actually do phage genome engineering. Specifically a type of negative selection, right? Because if it has the thing that if you supplied the things you don't want to see, the CRISPR-Cas13 is going to prevent those things from growing. And so then you'll only get the phages that have the changes that you want growing.
Yeah, the counter selection. So like where it just points out if you've got, I mean, I was thinking of Star Trek Discovery, where not Horizon Zero Dawn, where essentially there's, you know what, it's a complicated plot. I'm not going to go and make that reference, but essentially there are like a system to like destroy things if you kind of mess up your genome engine. So yeah. But yeah, I mean, if it was more common, then I'd be wondering, like, well, could you find a way to use an antibody to activate this system to just cause like it to activate in bacteria that are causing an infection, and then causing the bacteria to self-destruct, and at the same time, in order to evolve out of that, they'd have to make themselves more susceptible to phages. So it's kind of like a win-win situation. But that'd be an interesting concept.
Interesting. Yeah, you're imagining like using Cas13 as like, if you could deliver the system into like a bunch of different bacteria, it might like force them down an evolutionary path.
Yeah, like if you had like an artificial RNA like molecule that it was very non-specific in its binding, just bound to everything, and also bound to Cas, you could just like cause Cas to mess with like the host cells. But that's very much like a pie in the sky kind of idea.
Definitely. Yeah, so I think what's interesting about this paper is just the identification of this behavior for Cas13. We had previously talked about Cas13 as well, I think, in the phone diagnostic tool used for SARS CoV-2. And that was because, again, of this RNA binding ability. They didn't talk about, it took it out of the context of the cell. So it wasn't like Cas13 shutting down the cells upon seeing RNA. It's just Cas13 binding to RNA, right? And like for SARS CoV-2, an RNA-based virus, right, it can be useful in that way. So here's another use of Cas13, I guess, but within the context of the
Chris,
cell.
well, they can do absolutely everything. It's great.
It is definitely been such a, I guess, diverse and rich source of potential biotechnology tools.
Yeah. Speaking of like interesting biotechnology tools, we've got this paper called Mitochondria Transplantation Between Living Cells. Now this is something that I'm quite excited about because, because the first line of it I think says, let me see, like single cell surgery. Instantly, like, okay, this is going to be interesting. Like, what tools are they going to use to perform this single cell surgery? What are they planning? Yeah, that's.
You still have to wash your hands before you engage in surgery.
I mean, yeah, I presume so. I mean, they use atomic force microscopes to do this. I think, like where they've got that little like needle thing. Because we did talk about something recently, like where they're doing it with fungi, right?
Yep. Yeah, they were like extracting the cytoplasm from hyphae in order to grab the metabolites, right? So if you wanted to know like what biochemistry was going on in the specific part of the fungal cell, you could, pierce it with an AFM tip, suck up some of that fluid, and then do like metabolomics or something. And then they showed the reverse, that you could also inject into there. So this is like a really similar base technology, but now instead of fungal cells, we're looking at like larger eukaryotic cells, and playing around with sucking out. I guess they're making the hole a bit bigger as part of it as well, the aperture that they're on the cantilever.
They're testing out different forces to see what they can suck out. So firstly, the test is like, can they take out cytoplasm? And then they look at whether they can take out other, so endoplasmic reticulum, and then things that are attached to the endoplasmic reticulum as well, such as mitochondria, each using kind of different forces to try and get that needle to go into the cell.
Yeah.
And
well, like the forces are dependent on the aperture size, I think, is what they're saying. Like it's the hole that matters, how big the hole is that you're doing the sucking.
Definitely. And we've got like this has got lots of great videos of them piercing into cells. Nice. This flat bit is the kind of the surgical implement, which is essentially like this cantilever with a little needle on it, and the needle's got a hole that pokes into the cell, and then I think they use some sucking force that both like sucks up the lever and sucks up the liquid through it. So let's see you can sort of see the liquid going into it here.
Yeah.
And like I think there's another whole other slide where they look into it, if you look at this hole, you can kind of see a hole appear where stuff is just getting sucked into it, and that's
yeah, it's being pulled out of the focal plane, right? Like into.
So you can't see the thing that's doing the sucking, but you can see the things getting sucked up into it.
Right. Yeah. These structures are migrating towards a central point, and then they're being pulled out of the focal plane at that point.
Yeah.
Presumably into the tip.
And it's important to notice that the really bright spots are the mitochondria, and they're all like linked together at the moment in this kind of mitochondrial network that's working with the endoplasmic reticulum. It's getting like hoovered up by the cells. And the interesting thing is like you can see that they're all arranged in a line, but when you like start sucking them up, there's this moment where they get sucked up, but then like one gets sucked up, and then the structure kind of dissipates, and they kind of spring together into like these pearls on a necklace kind of vibes. So they become more spherical. So obviously there is some concern that are they damaging these cells during the process of sucking them up.
I mean, they're tugging on a membrane.
Yeah.
Right? It's interesting to think about, like, because it's suction and it's bringing up all this other stuff. Like maybe they could tear things. They could be pulling some protein interactions apart.
Yeah, that's obviously like a concern. And I think that what they find is that actually they don't have that much of an effect. So I think this image has like an image of calcium signaling. So if there was calcium signaling will happen if like the cell membrane is pierced, because most cell membranes have like a charge across them, and and once you break that charge, you can cause like calcium ions to like start spilling out the cell. But they don't see that, which is interesting. Yeah.
Okay.
So it seems like their actual process for extracting them, like it does a lot less damage than you'd expect to the cells. And so they did like two techniques to try it, and of course this was part of it. The other half was actually putting mitochondria into cells. So they've got like here they're going to put in like they've taken out mitochondria and they're going to put one back into the cell. So, and you can see I think it's shown up in red where they like start sticking mitochondria in, and you can see that the moment they like put in the cell, it starts integrating to the network of all the other mitochondria, and it starts growing up and becoming like part of. It starts fusing in with the rest of them. Yeah,
it's red in this video. I don't see any red.
Oh, right. Sorry. In the file. So it's firstly you see it's tagged with this protein called M-cherry, and so like you can see the M-cherry kind of vision on the far left.
I see.
And then in the middle is the original mitochondria, and then the fusion of them is. So actually, in the far right image, the mitochondria would appear white and red.
Gotcha.
Yeah.
I think also I'm seeing like a little bit of a truncated version, but I'm sure it's probably fine somewhere else.
Oh yeah. Apologies. Because we're communicating over Skype, and Danny can't necessarily see the YouTube video, so some things might get cut off. Yeah. Let me.
Cool.
Yeah.
Yeah, so I mean, like, it's kind of, this makes me think about, there's like chloroplast engineering inside of, in the plant world. People like make changes to the chloroplasts and then transform them into different cell types. I don't quite know why that's done instead of on the genome. I think it's an easier genome to edit.
Yeah.
This makes me wonder whether or not like people will do like, as a way of doing certain types of genetic engineering, like doing mitochondrial edits and then like inserting them into cells.
Well, that could be a really important thing, because the people get mitochondrial diseases, and one question for maybe curing them is finding some way to prevent. And this paper has kind of demonstrated that.
I mean, to every cell in their body.
Yeah,
that sounds
you're probably
crazy.
not going to do it for every cell in the body.
Like, with this,
but if it's something to do with, like, say, white blood cells, or something that you could fix via the bone marrow, then you could have it. So it's in the same way that, like, you irradiate and bone marrow and add in new like stem cells that can become the rest of them.
Oh yeah,
so
I could see that.
Potentially form like a kind of a stem cell treatment for mitochondrial diseases. Another interesting.
And at least to study.
Oh yeah, no. One interesting thing they found in the study. So there are a couple ways that you can actually get mitochondria into cells. So they tested out two techniques. One technique was where you could take a cell, pop it open, and just grab up a huge amount of mitochondria, and then give it to new cells. Which sounds like it'd be a much more like efficient technique than to poke it into to like take it out of one cell and put it directly into the other. But the actual like success rate of that was much lower than if you were to extract it using the needle and extract a couple of mitochondria using this needle technique and then put them into a new cell. So there is some other.
So this is the gentler way.
This is. I think there's a lot of like promise here. So this last one is in actual primary cells. I think they use human keratinocytes, where they implant them with these like mitochondria and see how well these mitochondria actually integrate with the network. Because another thing that they want to make sure is that these new mitochondria will just stay around. They won't drop out, and they don't need to add any selection pressure to keep them there, which again quite exciting. So yeah, I mean, like, I was talking to a professor like earlier this week who was saying that, are we like, wouldn't be, well, one of the questions we really want is to see whether we can do mitochondrial transplants. And I was like, well, actually, I read this paper just this morning about that very thing.
Nice.
So yeah, it was a moment where I, as a publisher who aren't historically known for knowing much about science, could use science knowledge.
Yeah, what was that? That's a plug for, you should read more papers with us.
Yeah, you should read more papers.
It's always, you never know what some tidbit of information is going to come in handy, especially like in science. So much is interdisciplinary these days. I feel like this paper is a little bit interdisciplinary for us as microbiologists because I do not think about mitochondria very much.
No, me neither. It's not. I kind of like accept them into the club of bacteria, but from a shared history and the fact they've got like the circular genome, and there's a lot of understanding between them. But yeah, they have defected to the eukaryotic side, so we might not have to. It might not be in our wheelhouse to cover, but it's interesting. I mean,
yeah, well, like it's kind of just like weird bacteria, ancient, endosymbiotic, right? They're like ancient offshoots, weird cousins.
Yeah, they still have like, bacterial secretion systems and other things to do with bacteria. So I'm still like counting them, but yeah, they've chosen a weird niche to live in.
All right. The next paper that we have is this asymmetric
peptidoglycan editing generates cell curvature in Bdellovibrio predatory bacteria.
We haven't talked about this type of bacteria very much. It's a weird one.
Yeah.
Like they are parasites, I guess. They call them predators because the bacteria end up predators of other bacteria. They are able to enter the outer membrane of other gram-negatives and like live in the periplasm.
Yeah, it's interesting because like they open up a hole, they live in the periplasm, and then they like send out all these toxic molecules that cause the prey molecule, the prey bacterium to like change shape, and then it digests the insides. And yeah, it's a really interesting organism.
Uh-huh. And you can imagine if there's a bacteria that eats other bacteria, the question has to be asked, can we make it eat the bacteria that we want it to eat? And some sort of therapeutic thing. So that's one of the interests. I mean, also just the sheer peculiarity of this lifestyle is what people are motivated by when studying, I think, Bdellovibrio, Bacterovorus.
Bacterovorus, yeah. It's and the interesting question they ask here is about why is it shaped like that? Why has it got this curved shape? So they try to find the gene that causes it, and they delete it to turn it into a straight bacterium. So I'm gonna hop over conversion therapy jokes. That would be insensitive. But turns out,
well, they also show that the straight bacteria aren't as good about like gaining entry to this
straight
periplasmic space.
Take a lot more time to get into them, and when they do, they don't fit in right. So like the curved the bendy bacteria are kind of they slot in quite easily, and you can see that they kind of are they kind of fit in, whereas the straight bacteria look a bit awkward and weird inside these.
Yeah. Which is, I wouldn't guess that. I mean, I always wonder, like, why do bacteria have all these shapes, right? Like in big organisms, the shapes have really clear functions, right? Like, oh, teeth and hands, and like these do things. But like the shapes in bacteria, like Bifido, Vibrio, cocci, like they're a bit more, yes. They're a bit more enigmatic as to like what those functions might be. So it's nice to see here, at least for this particular lifestyle, the curve has some effect in its desired niche.
Yeah, and they go in a bit more depth. They even like look at the gene responsible for the curvature and ask like, okay, why does it curve on one specific side compared to another? And they do some deletion studies to figure out that it's because of this specific part of the NFT, so not the NTF2 domain is required, not the NFT. We're not selling that yet. But...
Yeah. NTF2 in an enzyme that modifies peptidoglycan, right?
So it actually.
Yeah. So I was going to say, so peptidoglycan is like it's quite an important molecule in the bacterial world. It creates a lot of structure, and there's like a lot of it's essentially like a giant carbohydrate, right? Sugar carbohydrate that or peptides too. There's also protein in it. Anyways, it's one big macromolecule that covers the entire cell. There's a lot of different enzymes that interact with that and change the properties of the peptidoglycan. And so this particular domain that Faz, you're speaking about, like is catalyzing a specific chemical change in the peptidoglycan that allows it to adopt this shape. They think it's like weakens like something or strengthens one of the edges so that maybe the pressure or whatever of the internal cytoplasm is like pushing out the Vibrio shape.
Yeah. So yeah, it's kind of a neat thing that we don't often think about in too much detail because usually, like, we're not at the same scale of the bacteria, so you wouldn't be able to see, like, that the curvature allows these bacteria to actually fit in to their niche. So they infect bacteria and make them spherical, and because of that, they can fit in with their curved shape. Whereas when they're straight, they don't fit in right, and so it doesn't.
Yeah, well, they poke out of the ends, right? Like it's kind of like it doesn't work in the biophysics of trying to fit this bacteria into another bacteria. I guess the other thing I was going to say is that this, I read in the discussion that the authors say that this mechanism of like Vibrio shape is not like other Vibrio shape mechanisms. Right. So like in other Vibrio, they have like an internal structure that forms their Vibrio shape. But yeah, for this one, it's like about it's interacting on the peptidoglycan outside. So, many different ways to achieve similar results. That's the story of biology as well.
Fascinating. And I think that brings us to our last paper, Candida auris on apple and clinical significance. So,
so yeah, Candida auris is this. There were at least at one point a lot of scary articles saying that like there's this new yeast that's showing up in hospital infections. It's incredibly drug resistant. What are we going to do about it? And so in this paper, the authors are following up on an observation that said, oh, actually, Candida auris comes from the marine environment. They've found like strains of Candida in the oceans, but those Candida don't have antifungal like resistance genes. So something happened along the way, is the hypothesis. The ancestral Candida somehow picked up like resistance to different antifungals and then made its way into hospitals and is just wreaking havoc. And the authors hypothesize that this could be the place where they could have picked up these antifungal genes is on the surface of apples because apples, many fruits have yeast on their surface. And when apples get stored, they get stored sometimes with some fungicides. So this could be like the selection pressure. Essentially, our apple industry could be breeding or could be increasing the diversity of genes inside of Candida auris that then become specialized inside of our hospitals with not good results.
Yeah, this is interesting because it kind of like, because with apples, part of the thing that I'm thinking about here is that they often get stored a long time, like because because a lot of people like to eat apples out of season, and because of that, we have to store them in an unripe state, usually on like container ships where they're being shipped to their destination, and so there are all these preservatives we need to make sure that they don't get infected. And now it's
I will say it's not just because people eat apples out of season. I think it's one of the strengths of the apple crop is to be able to be stored. Like I believe like there's even, I've just been going to the community garden a lot recently, maybe because it's a nice outdoor activity you can do. And like there are specific breeds of apple that were like bred to be stored. So like they don't taste good right off the tree. Like they're intended to like sit in storage. And like apples are also like incredibly hardy, and they usually coat them with wax. Is one of the ways that they help, like so they don't lose all their moisture in storage.
Yeah, no, I remember I think there's an entire module on like food preservation during my microbiology degree because this like kind of stuff, this thing that we have to like food is really important. So it's an important part of our system. Now it's like linking up to health. There's a lot of like angles here.
So they look at Red Delicious and Royal Gala apples that were picked in India, and they swab the surface of apples. They get a whole bunch of yeast sequences, and then they put them into big phylogenetic trees and compare them to like known disease-causing isolates and so forth. And they find that there is similarity between some of the strains that you find in hospitals and these strains found on apples. And furthermore, it's not just in India that you can find this like relation. It's also like in other countries. So like other countries have yeast that cluster phylogenetically speaking with the yeast that they're finding on these apples. So and there is low levels of fungicides being used in the waxes on these apples and so forth. And so again, that all this information kind of comes together to suggest a story, which we've talked about before, about One Health, right? Where all of our interlocking systems are contributing to human health. Because microbes, they are everywhere, and they can transfer between all these different interlocking systems. They can move their genetic diversity between the systems to find the niche that they're going to be successful in. And one of those niches is disease. So being able to pick up, fungicide-resistant genes in one environment, perhaps the apple environment, could lead to the genetic diversity required to pick up more serious resistances in hospital-acquired strains. And again, that's just a hypothetical framework, but this study in picking out the relationship of the yeast on apples to other yeasts suggests that this could be one of the things contributing to global health in this way.
And I want to point out that they did find more diversity in the apple storage than they did in the hospitalizers, right? So that, because the obvious kind of counter argument here is say, well, these things can operate two ways. I mean, people can spread Candida auris to apples, but that kind of the fact there's more diversity, we were seeing strains of Candida auris that are resistant that aren't related to hospital that in the storage, that kind of seems to point to a one-way way of things moving.
For sure. I don't think it's super strong evidence for that one-way nature necessarily. I mean, it does make a lot of sense that there could be, but you could also imagine if it was the case, it just seems weird, right? That a disease-causing strain in humans would find its way into apples and then radiate into a lot of different types. So more likely that this is the source of the diversity that gets specified.
You say that, but then actually, in that environment where in both environments, it's advantageous to be evasive to antifungals. So
that's true.
Yeah. Both hypotheses can happen.
You can't say this stuff just from looking at the genetics. I mean, this is, we're talking. This has relations to how we talk about SARS CoV-2 spilling over in different ways too, right? How do you know it came from us when do you know the directionality of things? Usually it means you need a lot more sequences. You need to see all of the intermediate stages.
Yeah, we need more science.
A constant refrain from every scientist.
Yeah. I think that brings us to...
All right. Well, that brings us to the end. So we hope you enjoyed it. Join us next week for more cool microbiology news.
Yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter @microtwjc. @microtwjc. We both believe that peer review is a process that we can all participate in, so that we've hoped that you had a good time listening to us ramble on about microbiology today. Thanks for all the comments in the chat, and if you found something clear, if you want to add something more or found something unclear, just let us know in the comments.
Yeah, thank you for the comments. They've been making me laugh all through this session, so I'm quite happy about that. It's been a pleasure chatting with all of you, and especially with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Goodbye.